Hello all, welcome to Simplified Bio by Sidhu. This is your Simplified Biology teacher. So in previous video we have discussed about photosynthesis. So how exactly photosynthesis will occur and what are all the things that will happen. All these things we have discussed. So in this video, so let us discuss about the raw materials, whatever the things we have discussed in a photosynthesis like carbon dioxide, sunlight and chlorophyll. So whether these are essential or not. To prove that, so we have some experiments. So they may ask you in the exam for 5 marks, which are very important. So in a simple way, I'm going to explain it. Okay, so which we will explain in step by step, so that observe very carefully and write it down. So before going to that, if you are new to our channel, please do subscribe and hit the bell icon to receive the notification, and also share to your friends who are in need. So let us start with our session. So first experiment is that, so experiment to show that chlorophyll is essential for photosynthesis. Always while writing the equation, you will write chlorophyll absorb the sunlight, isn't it? So how to prove that? So with this experiment. So what is the aim of this experiment? To show that chlorophyll is essential for photosynthesis. Okay, to show that chlorophyll is essential for photosynthesis. So what are the materials required for this photosynthesis? To this experiment. So variegated healthy potted plants with variegated leaves. So variegated leaves in the sense, so where you will find these kind of leaves. See if you observe. So patchy leaves. So chlorophyll will be present only in few parts of the leaves. Okay, so like this. These kind of leaves you might have observed. Okay, so patchy leaves. So example for this leaves are crotons as well as folios and even Tragacanthia. So these are all comes under variegated leaves. See, some leaves you may find along with the green color, the leaves also you may find yellow or red in color. Okay, so such kind, or even sometimes white patches will also be there, and such kind of leaves can be called as variegated leaves. And you have to take such kind of one plant with such kind of leaves, and then so you need alcohol. Okay, so and then Bunsen burner, iodine, and then Petri plate. So these are the materials required to conduct this experiment. Okay, so let us move into the procedure how to conduct the experiment. For the first, you should take the plant and whatever the variegated leaves. So you should take the plant and you should place the plant in dark room for 3 days. Okay, what you people are going to do? So you should take the plant and then you are going to keep that plant in a dark room. Here question arises, why we should keep it in a dark room? So why can't we conduct directly? Okay, so if you keep it in a dark room, so that whatever the starch which is already produced, it will be consumed. Okay, for the next 3 days. When you keep it in a dark room, the sunlight will not be available, so that they will not be able to produce starch, so that whatever the starch which is already present, so they will be utilized. Okay, so that is what you will call de-starch. Okay, so you should keep the plant in a dark room for 3 days to de-starch that. Okay, so next after de-starching, so once you remove the starch which is already present in that, so what you are going to do? So same plant you are keeping in the sunlight for 4 to 5 hours. So now in this plant, so whatever the chlorophyll which are not present area, in that area no photosynthesis will occur. So when you keep this in the sunlight for 4 to 5 hours, So whatever the chlorophyll are present in that area, photosynthesis will occur. Okay, so then you should take one leaf from that plant and then you should conduct test for starch. Okay, so this is the procedure that you have to do. So usually some books how to do test for starch also they have provided, it is not required. So even if you write, no issue. So how to do test for starch? So first you have to take the leaf and you have to boil it in a water bath, and then so same leaf you should keep it in a alcohol and then place it in a water bath. So indirectly you are boiling in alcohol now. Why you are not boil in a alcohol directly with the flame? So everyone knows that it is highly inflammable. Alcohol catches fire. So to avoid that, we are keeping in a water bath. Okay. Next, so see, so when you boiling it in a water bath, so the color of alcohol turns green. It will turns green. So why, can anybody tell me? So it takes up all the chlorophyll which is present in that, so that the plant looks almost transparent or white in color. Okay, so next what we are going to do, so we will add, we should take that in a petri plate. So we should take that in a petri plate. Now I am keeping this leaf in a petri plate and I am adding iodine. I am adding what? Iodine solution to it. Once after adding iodine solution to it, what is going to happen? So I think you got an idea. So wherever the green patches were present in that particular leaf, so that particular leaf will show blue-black color. Okay, so observation. So only the parts which were green previously, that means wherever the green patches were found, all these green patchy area will show blue-black color. The remaining, where you will not find the green patchy area, either you may find red or you may find yellow or you may find white. In that particular area, you will not be able to find any kind of blue-black color. Can you tell me why? Because there is no chlorophyll here. Okay, so hence in the conclusion, so we are going to write, so this experiment shows that chlorophyll is very much essential for the photosynthesis.